Assalamu alaikum. I am Dr. Uruj Aziz from Upper College Karimabad. I did my PhD under the supervision of Dr. Noor Saher and Dr. Mustafa Kamal from University of Karachi. My today's topic of presentation is phylogenetic and phylogeographic structure of crab of genus Macrophthalmus from intertidal habitat of Pakistan. Introduction. The branchiodont crabs are a well-studied group because of their morphological and ecological versatility. Among them, the crabs of genus Macrophthalmus are highly diversified crab. They have Indo-Pacific distribution, including tropical and subtropical countries like Australia, Taiwan, Kuwait, India, Pakistan, Persian Gulf, China, etc. These are the most important intertidal species. As a deposit feeder, they feed upon dead organic matter, result in nutrient recycling. They are important source of food for predators. Their biodistribution activities help in nutrient or material recycling in the sediment, and they are also considered as bioindicator species of heavy metals that are present in mangrove intertidal environment. As I said earlier that these are highly diversified crab, but genetic studies on this group has not received much attention until recently. Genetic diversity is the sum total of variation or the amount of genetic variation within and among individual of population or a species. Genetic diversity can be calculated interspecifically or intraspecifically. Genetic diversity not only provide phenotype of an organism but also provide a raw material for natural selection and adaptation towards the changing environmental condition. These things ultimately results in speciation. Now the aim and objectives. The aim of this study is to evaluate the molecular variability within the species of genus Macrophthalmus and to evaluate the level of intraspecific population structure among its distributional range. Now the material and method. Random samples of these crabs were collected from different sites by handpicking method. These sites include Sanskrit, Sonari, Somyani, and Kurangi. Just after collection, the specimens were kept on ice and then transported in laboratory. The total DNA was extracted from muscles that are present on the periopod with the help of a DNA kit that is known as SkyGene. Then after the extraction of DNA, the PSCR technique has been used to amplify the DNA template. During current study, partial cytochrome oxidase gene has been amplified using PAMR and thermocycline profile as given in table 1 and 2. Now result and discussion. For morphological identification for the crab genus Macrophthalmus, different available keys has been used that include Termesi Anwani 1996, Barnes 2010, and Motherloo 2011. According to these keys, total 5 different type of species have been reported, including Macrophthalmus depressus, Macrophthalmus levis, Macrophthalmus sulcatus, Macrophthalmus dentipus, and laterali. Now the habitat. This slide shows some clicks of sampling sites that include Somyani, Sonari, Sanskrit, and Kurangi Tweed. Positive amplicons were sequenced from Equagen Korea. These amplified sequences were submitted to NCBI as shown in Table 3. Megasoftware has been used for statistical analysis. New sequences and sequences available on NCBI database from Indo-Pacific region were downloaded for further analysis and evaluation. During current study, sequences were aligned by cluster W. approximately 604 base pair of cytochrome oxidase gene were used for the construction of phylogenetic tree. The studied model for tree construction is GTR plus G plus I, and the nucleotide were AT-rich as reported previously. Now the next slide. Maximum likelihood method and neighbor joining method has been used to evaluate phylogenetic relationship. During this phylogenetic relationships, swimming crab of genus Porcinus has been used as an outgroup. COI sequences of Macrophthalmus sulcatus and laevis has been sequenced for the first time from Pakistan. Similar is the case for Macrophthalmus laevis and Macrophthalmus dentipus. For Macrophthalmus depressus, data is also available from India, and for Macrophthalmus laterali, data from Pakistan, China, and Taiwan has been used during the current study. Now the next slide. Maximum likelihood tree suggests that Macrophthalmus sulcatus shows closer association with Macrophthalmus laevis, while Dentipus and Macrophthalmus laterali are genetically closer to each other. Now the next slide. This slide shows neighbor joining tree and gives similar result as previously, that is then sulcatus and Macrophthalmus laevis are form a closer group, while Macrophthalmus dentipus and laterali are closer to each other. The results also show that Macrophthalmus depressus are closer to Macrophthalmus dentipus and laterali, while Macrophthalmus sulcatus are distantly related to laterali and dentipus group as compared to depressus. Now the next slide. Intraspecific genetic variation within different species. For inference of intraspecific genetic variation, genetic data is not much available. For example, for Macrophthalmus sulcatus, no data is available from its distribution range except for a subspecies, that is Macrophthalmus melacensis from Taiwan. Same is the case for Macrophthalmus laevis and Macrophthalmus dentipus. Now the intraspecific genetic variation within Macrophthalmus depressus. Data is available from Pakistan and India. From India, only a single sequence is available, while it is separatory placed in phylogenetic tree as we shown in our previous slide. For the formation of true picture, we require some more sequences so we can depict a better result. Now the intraspecific genetic variation in Macrophthalmus laterali. Three sequences were available, that is from China, Taiwan and Pakistan. Tajma D test and haplotype diversity test show that there is no genetic variation among them. Studies show that those animals which have planktonic larvae have high dispersal capacity and level of genetic flow, which reduce population genetic structure as reported in this case. Now the conclusion. The wide distribution pattern of crab of genus Macrophthalmus and their larval dispersion help us to investigate the genetic variability, which is better for the comprehension of biogeographical processes, population differentiation, and biodiversity of the group. These are some references. Thank you so much.